This video is a summary of the first part of the carbohydrates topic for A-level biology. In this, we're just going to look at carbohydrates in general and the monosaccharides and disaccharides. There's a separate video for looking at the polysaccharides. Carbohydrates are a really important class of biological molecules, and as the name suggests, they contain carbon, hydrogen, and also oxygen. Because as you know from GCSE chemistry, 8 is a chemist's secret handshake to say this molecule contains oxygen. And those are the only 3 elements that we're going to find in a carbohydrate molecule. But we can actually go one further than that. Rather than just saying these are the elements that these molecules contain, we can give them a general formula. So you know from GCSE chemistry that a general formula is basically a molecular ratio that tells you about the different elements that are in a particular class of compounds. So here we can say that the carbohydrates have a general formula of CxH2OY. And X and Y can sometimes be the same number as each other, but they can also be 2 different numbers. So what we're saying is that within a carbohydrate, you're always going to have twice as much hydrogen as oxygen. And then sometimes you'll have the same amount of carbon as oxygen if X and Y are the same number. So for instance, you already know about glucose, in which you have 6 carbon atoms and you also have 6 oxygen atoms, but also you could have a different number of carbon atoms. Carbohydrates are also known as saccharides, and we can break that down further depending on the size of the carbohydrate. So again, you know from GCSE chemistry that we have polymers, which are very long molecules, and they're made up of smaller molecules called monomers. So we have monosaccharides, which are our smallest, simplest sugars. So these include things like glucose, which you already know about from respiration, fructose, which is found in fruits, galactose, which we'll get to in a second, that's quite important in one of the disaccharides, and also things like ribose and deoxyribose, which are found in nucleic acids. Now, if 2 of these monosaccharides join together, they can form a disaccharide. So di means 2. So you've probably heard of lactose in the context of people being lactose intolerant if they can't have dairy products. And that's because lactose is the disaccharide that is found in milk, and it's made from glucose and galactose. And then we've also got another disaccharide, which is sucrose, and that's what you think of as table sugar. And then if we bond lots of these monosaccharides together, then we can start to produce polysaccharides. So these are these much larger storage carbohydrates which are used to store energy long term, because they're so big that they are no longer soluble, and therefore they're not going to affect cells in terms of osmosis and drawing extra water into the cells. So you've probably heard of all of these from your GCSE studies, but glycogen is the one that we find within animals, sort of stored in the muscles and stored in the liver. And then starch is a plant's main storage carbohydrate, and then cellulose, of course, is a structural carbohydrate, and it's used to make up the cell walls of a plant. You should also know that glucose, fructose and galactose all have that same molecular formula of C6H12O6, but they differ in their spatial arrangement of those atoms. So to give you a closer look at these different types of carbohydrates, on the left-hand side we have glucose, which is an example of a monosaccharide. And then in the centre we have sucrose, which is a disaccharide, and you can see there that even though it has 2 subunits, they're different to each other. So on the left-hand side we have glucose again, and on the right-hand side we have fructose. And then on the right-hand side of the screen, we've got a polysaccharide that you can see I've labelled amylose or amylose. And you might think, well, that's quite a familiar name, but I don't think I've ever heard of it. And the connection you're probably making is with the enzyme amylase or amylase, whichever way you prefer to say it. And that's because amylose is one of the 2 different carbohydrates that make up starch. So you learnt at GCSE that amylase is the enzyme that breaks down starch. This is what it's named after. So why do we care about these carbohydrates? Well, very early on in your school career, you learned that simple sugars, so monosaccharides and disaccharides, provide a source of energy to allow cells to do all of the things that they need to do. And then more complex carbohydrates like starch and glycogen are used as stores of energy. And then later on, they can be broken down to produce those simple sugars, which are then used for respiration and to provide a source of energy. You also learnt that in plant cell walls, there's another carbohydrate called cellulose, and this is an example of a structural carbohydrate, and there are others as well, such as chitin. Now, one thing that you may not know about carbohydrates is that they're very important in cellular recognition and in signalling. So things like hormones, when they come and bind to the outside of a cell, will typically be bonding to something called a glycoprotein. And you're probably looking at that word and thinking, well, surely a glycoprotein is a protein. And you'd be right. But the glyco bit tells you that it's been modified to have a carbohydrate attached to it. So glyco is a prefix that indicates that something is to do with carbohydrates. For instance, when we start talking about those disaccharides, we'll mention glycosidic bonds. And then the other thing that carbohydrates are important for is that they are a base unit that is required to make other molecules, such as the nucleotides that make up DNA and RNA. You need to know the details about 5 different monosaccharides and 3 disaccharides. So we'll start with monosaccharides, which are glucose, fructose, galactose, ribose, and deoxyribose. And you'll notice that these are split into hexoses and pentoses. And you can probably figure out, based on your maths knowledge, that hex refers to 6 of something and pent refers to 5 of something. So this is actually the number of carbons that are found in these molecules. So you already know that glucose is C6H12O6. So as you can see there, we've got 6 carbons, and that's going to be true of the rest of the hexoses as well. And then the pentoses only contain 5 carbon atoms. Despite having the same molecular formula, fructose, glucose and galactose have different sweetnesses, and this is based on the different arrangement of those atoms in space. So fructose is sweeter than glucose and glucose is sweeter than galactose. Next, we've got our disaccharides. So remember, di means 2, so they're going to have 2 subunits in them. So maltose is just made up of repeating units of glucose. So one glucose bonded to another glucose. And if we carried on bonding those glucoses together, then eventually we could make some starch. Then sucrose, we've said, is largely found in fruit, and this is made up of fructose bonded to glucose. And then finally, we've got lactose, which is the disaccharide that makes milk slightly sweet, and this is made up of glucose together with galactose. Glucose is a really important carbohydrate because of its role in aerobic respiration, but you may never have thought before about its structure. So to start with, glucose is an example of a hexose monosaccharide, and we describe it as a hexose because it contains 6 carbon atoms. It has 2 different isomers. So if you don't take chemistry, isomers are molecules that contain the same atoms, but they're arranged in a different way. So both of them start off like this. You can see we've got a sort of hexagon shape, but one of the corners of that hexagon is not a carbon, it's an oxygen. And so actually the 6th carbon is coming off on this little group that comes off the top. Now we number these carbons starting from the right hand side of where that oxygen is. So we have carbon 1, 2, 3, 4, 5, and then the one that's not included in the ring is carbon 6. Now you can see here that on the right hand side I haven't actually finished off the molecule. I've got 2 bonds coming off carbon 1 that currently don't have any atoms on them. So in alpha glucose you have a hydrogen at the top and a hydroxy group at the bottom, whereas in beta glucose those 2 are reversed. It's also important that you know that glucose is polar, and this means that it's going to be soluble due to the formation of hydrogen bonds between all of these hydroxy groups and also the water molecules. Time for a quick progress check, so pause the video and see if you can work out the correct answer to this question about the structure of glucose. Well, we can start off by thinking about the molecular formula of glucose and using that to rule out some of these answers. So we've got a carbon to oxygen ratio of 1 to 2. Well, we can see here we've got 6 carbons and 6 oxygens. So answer A cannot be correct. And then we've got a carbon to hydrogen ratio of 1 to 2. So we've got 6 to 12. So that is correct. So B could possibly be a correct answer. And then for C, we've got a carbon to hydrogen ratio of 1 to 1. So we've just said that that's not right. And then finally, a carbon to oxygen ratio of 1:1. So that is right. So we know that our correct answer is either going to be B or D, and we can rule out A and C completely. So now we actually need to think about what does this molecule look like. So if we count up our different hydroxy groups, our OH groups, we can see we've got one here, one here, one here, one here, and one here. So 5 of these carbons have got hydroxy groups associated with them, but our carbon that is here does not have one, and we've got an oxygen that is not part of a hydroxy group, it's just part of that ring instead. So we've only got 5 hydroxy groups, and therefore our correct answer is going to be B. Can you also describe the difference between alpha and beta glucose? So the important thing here is about the position of that hydroxy group, because in alpha glucose, that group is below the carbon ring, but in beta glucose, it's above. The next monosaccharide we're going to concentrate on is ribose. So ribose is really important because it's one of the components that makes up RNA, that nucleic acid that is responsible for transmitting information from the DNA out of the nucleus and into the rest of the cell. We describe this as a pentose sugar because it contains 5 carbon atoms. But just like with glucose, where we saw that there were 5 carbons in the ring and then 1 off to one side, we have the same situation here. So we have 4 carbons that are part of the ring and then a 5th carbon that is making a bond away from carbon number 4. So now that we've met some important monosaccharides, we need to think about how these join together to form disaccharides. A condensation reaction is a particular type of chemical reaction where a large molecule is formed from 2 smaller subunits and a small molecule is lost at the same time. If you studied GCSE chemistry as opposed to GCSE combined science and you learnt about esters, then you'll have already met this example of a condensation reaction. So here we've got an alcohol reacting with a carboxylic acid to produce a water molecule and also an ester molecule. But condensation reactions are also really important for making lots of the molecules that we study in biology. Here we've got 2 alpha glucose molecules which are going to bond together to form a larger molecule of maltose with the loss of a small molecule of water. So this bond is going to form between carbon number 1 on my left-hand molecule and carbon number 4 on my right-hand molecule. And so you can see we're going to lose that hydroxy group on the left-hand glucose, and we're going to lose just the hydrogen from the hydroxy group on the right-hand glucose. So that's where our water molecule is coming from. And so we're going to form this larger molecule of maltose where the 2 glucose have joined together. And because that bond is happening between carbon number 1 and carbon number 4, we're going to refer to it as a 1:4 glycosidic bond. So a glycosidic bond is a covalent bond, it's just specifically one that is forming between sugar molecules. And this is sometimes also referred to as an oxygen bridge. We started out with 2 molecules of glucose, which we know have the formula C6H12O6, and that's going to be true for glucose and galactose and fructose. And then the disaccharide that we've made, we could count up all of the individual atoms, but actually we know that conservation of mass is a thing, so we're still going to have our 12 carbon atoms, 24 hydrogen atoms, and 12 oxygen atoms. But 3 of those have been used up in that 1 water molecule. So what we're left with for this maltose and for the rest of our disaccharides is C12H22O11. So pause the video and see if you can write down how does this glycosidic bond form in maltose. So we know that this is a covalent bond that is forming between 2 glucose monomers, and specifically between the hydroxy group on carbon 1 of 1 molecule and carbon 4 on the other molecule. It's an example of a condensation reaction with the removal of a water molecule, and the 2 glucose monomers are now linked by an oxygen bridge. We've said that condensation is a reaction that joins 2 smaller molecules together with the loss of water. So you can think of hydrolysis as being the opposite of condensation, because here we're adding water and we're using it to split apart a larger molecule. So this is used to split up some of those larger storage carbohydrates in order to release glucose for use in respiration, and it's catalysed by various enzymes. So as you learnt from GCSE, generally speaking, enzymes are named after the thing that they can break down. So amylase breaks down amylose, maltase breaks down maltose, and lactase breaks down lactose. One final progress check about carbohydrates. So pause the video and see if you can identify the option that correctly describes the structure of lactose. Our first option is that lactose contains phosphodiester bonds that are broken by a hydrolysis reaction. So we like the idea of being broken by a hydrolysis reaction, but the phosphodiester bonds are wrong. We know that actually we're looking for glycosidic bonds, so we can rule out option A. Option B, lactose is made up of fructose and galactose. Well, the galactose is correct, but the fructose is not. C, the molecular formula of lactose is C12H24O12. Well, that would be true, but we know that when lactose forms, we're going to lose that water molecule. So actually, this should be H22 and O11. And then that leaves us with lactose contains glycosidic bonds, much better, that are formed by a condensation reaction, and we like that as well. So our correct option is option D. Thank you very much for watching, and I hope you're now feeling a bit clearer about monosaccharides and disaccharides. If you did find the video useful, then don't forget to like, subscribe, comment and share, and I'll be back soon with some more A-level biology videos.